So any of these purple things, they're all proteins, right? The purple things are all proteins. If it goes through the membrane, it's an integral protein or a transport protein. This, we know the green antennas are carbohydrates. If it's attached to the big purple thing, it's a glycoprotein. If it's attached to one of the yellow balls with tails, right, it would be a glycolipid. So here you can see the glycolipid. So the heads and the tails, those are your phosphate and fatty acids. So they're the phospholipids. This is only on an edge, so it's a peripheral protein. Okay. And then integral we already did. And then the rings, like 4 sets of rings, are your cholesterol. Okay. Moving up. So here is how these look. A channel protein. Oh, that was tough. It's a channel. So we're going to talk about channel proteins today. They allow things to pass through. So the rest of this is mostly from Friday. Cholesterol prevents them from sticking together. Phospholipid heads, that's the hydrophilic portion attracted to water. Fatty acid tails are the hydrophobic portion. They repel water. Glycolipids, any of those glycoportions do have to do with cell recognition. Okay, I think that was all of them. Okay, any questions on any of those? Want any repeats? You could use that and make flashcards for studying. Gives you a set of vocabulary. Oh, good. Okay. So now I'd like to do another task. So I'm going to run this more like a class today where we're actually like accomplishing a handful of things. So we might go a little longer, but you will still have a full hour for your flex. So I'm going to share a different screen now. I want to go to your test. Okay. So we got this done. We went through our test, and now we're going to get into new content. So you can open up in your Google World if you'd like to follow along the test, or I mean the PowerPoint. So we're going through membrane transport. We reviewed membrane structure. At the end, the second document is just the single slide self-check, okay? As homework today or as our flex work, and you'll have plenty of time in the 1 hour that you have a short virtual lab to do. So play the videos out. It's going to show you what happens. I forget, the video might be 5 minutes. And then you're just going to answer questions based on what you observed in the happenings of the video, okay? So that'll be later. So I'm moving into the PowerPoint. Are you guys ready to roll with me? Okay, nobody's saying no. So I like to put it in presentation mode. So we're going to talk about how things get into and out of the cell. Last week we talked about the fact that we need to take in nutrients, oxygen, water. We need to get rid of waste, toxins, things like that. So for the sake of time, I'm going to skip this animation. It's a good like animation, simulation, if you will, showing you how molecules move through different forms of passive transport. You might, when you're reviewing the PowerPoint, you might go through and click that yourself, okay? So this lecture is about passive transport. And when we say passive, we say we don't need any energy. That's what it's referring to. So chemical energy in the form of ATP is not being used. It does require a concentration gradient. So we need to understand 2 words there. Gradient, which means the difference between 2 sides, and concentration, which is how much stuff is in a given area. So if I had a teaspoon of sugar in a cup of water, it would have a teaspoon per cup, say, concentration. And then if I had a second cup and I put 2 teaspoons in, that's a double concentration, right? That's more concentrated sugar solution. So it has more sugar in the area. So the difference between those two was a 1 teaspoon difference, which is a 50% grade, the difference between two sides. So on the highway, you might see a sign, especially if you're out west maybe, and it might say like a 12% grade, which is a really steep hill for your cars. So it gives you a warning. Active transport, we're going to talk about a different day, but we'll compare them now. It requires energy. That's why it's active. Passive, you just let things happen. If you're actively involved, you're participating, right? So you need energy. It will go the opposite direction. So it takes no energy to go down a hill, but it takes a lot of energy to go up a hill, right? So the active transport goes against a gradient, so it requires energy. And then bulk transport is more about the cell engulfing, kind of like that endosymbiotic theory we talked about, where the membrane is taking part. It's not things moving through. So today, talking about passive transport, we introduced diffusion, which is simply the movement of molecules from a high to a low concentration. You should have that definition memorized. I'm going to ask you it again in AP Biology. I'm going to ask you it again in anatomy. So this word should stay with you. So things move from a crowded area to a less crowded area. Here you have this lump of sugar. The molecules are very closely packed. Atoms are in constant motion. So are molecules. So they're going to hit each other, and it causes them to ricochet off, which causes them to spread out. So diffusion, things will move from that high concentration here to a low concentration. Out in the water, there was no sugar to begin with. So it moves out into the water space until all things are equal. So these molecules are evenly spread out. So this requires no assistance of any kind. A facilitated diffusion is still going from high to low, but to cross a membrane, it needs a carrier protein of some sort. So these are categories of passive transport. So I'm making my concept map, right? Osmosis is also a form of diffusion, but it's specifically water. It's not talking about sugars or dyes or proteins or anything else, just water. Okay, so diffusion is caused by the process of Brownian motion. And Brownian motion is that ricocheting I talked about, okay? So the molecules bounce off of one another. So I'm going to play this video just for a little bit, and she's going to talk. We don't care about her. What I want you to notice is just look at the particles that are in this glass. You should see them bumping into each other, and that causes them to spread out. I tried to turn my sound off last time, and I ended up messing up.
Brownian movement. The continuous zigzag motion of fine particles suspended in a gas or a liquid is called Brownian movement. It was discovered in the year 1827 by the botanist Robert Brown and is named after him. For example, let us observe a beam of sunlight entering a dark room. You will find that the dust particles are constantly moving in a haphazard manner. What makes dust particles move continuously in air? You already know that air is a mixture of gases, and gas molecules are always in a state of free random movement. These molecules hit the dust particles from all directions and make them move in an irregular manner. Similarly, if pollen grains are placed in water, they do not remain at rest, but move about continuously and randomly. This is also an example of Brownian movement.
I didn't want to play that whole thing out, but my controls pop in the way of the stop. So I really liked the demonstration about seeing the dust particles in the light, because I bet we have all seen that. Like, we don't see particles inside of the water moving, but you've seen dust like from a movie projector, and you can see the dust that's in the air and how it's hitting each other. So this is showing you that ricochet, okay? So a common example of diffusion would be the gas that enters our lungs. It has to move across our membrane. And it's the bouncing, that causes that to happen. So Brownian motion. So I'm not going to play this one out because that one played so long. This is a good example. So if we were in class, this lecture would have all kinds of demos. So if you put food coloring in water, that would be the high concentrated area. And then, you know, if you let it set for a while, it's going to diffuse. It's going to move out to fill the space. So it's moving from a high area to a low area, where it's highly concentrated to a low concentration, until they are all equally distributed. So equilibrium is equally distributed. It's dynamic because even though they're equally distributed, they don't stop moving. They keep moving. So it's like staying the same while changing. Again, I'll let you click on that link. I'll let you play this video out. In the first animation, there wasn't a membrane involved. This one has a membrane, so you'll see how it moves from this high concentrated area. Particles will bounce into each other, causing them to spread out. Anything that speeds up the rate of bouncing is going to speed up diffusion. So if they're more closely packed together, or if the temperature is warmer and they're moving more quickly. So in this video, you'll see them move through the membrane as well. So temperature plays a role, of course, because molecules are moving at different rates. So notice how the dye is diffusing at different rates, spreading out to fill the container in an equal distance, okay? Clearly the hot went really quick. The slow or the cold is still sticking together. Molecules move really slow when it's cold. That's why we have to warm up before exercise, because it makes it so molecules inside of our body are moving more quickly, so we have better reactions. Okay, the warm, somewhere in between the two. So the rate of the movement of the molecules causes those ricochets to happen more quickly or more slowly. Putting pressure on it compresses them, makes them more concentrated in an area, which is going to make the collisions more common, more often. Concentration gradient, same thing. A teaspoon of sugar versus 2 teaspoons of sugar in the same space. The 2 teaspoons of sugar molecules are going to hit each other more often. So all of these affect the rate of diffusion. You can speed it up, you can slow it down. Facilitated diffusion occurs with a carrier protein. So before we just talked about diffusion and it required no outside source. So we just refer to that as simple diffusion. Now we're talking about facilitated diffusion. Facilitating is assisting, right? So I'm trying to assist your learning. So this is, I'm trying to facilitate your learning like a protein facilitates diffusion. So if things are too large, like a protein is too big to fit through the tiny little pores between the lipids, right? So it's going to go through what's called a carrier protein. These on the end, this is a carrier protein. So the carrier protein moves large things, and it changes shape to move it from one side to the other. So I see it changing shape there. The other type of protein we looked at were channel proteins. So notice it's a tube. So this allows polar molecules to slip through. So things like sodium and potassium, they have a charge on them. So does chlorine. That charge is going to be repelled by the inner membrane, so they wouldn't be able to pass through. So we have to protect them from the charges and provide a tube for that ion to pass through. Okay, I'll just play a short bit of this. It's going to be an animation.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor, which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, This protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor, which helps molecules move across the membrane.
Okay. So that sounds like a good test question. What's common between facilitated diffusion and diffusion, and what's the difference between them? Okay, so facilitated diffusion requires proteins. Osmosis is a form of diffusion, but it only refers to water. So it's the movement of water across a semi-permeable membrane. So to save us time, I'm not going to play this video out. You guys can watch it later. But you're going to see the components of the membrane, and you'll see how the water molecules are moving across. So moving from water's high to water's low concentration. So in this case, I have a concentrated salt solution. So there's a lot of salt in this tube. There's no salt in the water. So to make the two sides equal, water, if I'm only talking about the movement of water, water can't come out of this tube and into the beaker and then they become equal, right? Water's going to have to go from the beaker into the tube. So water moves into the tube and we see that rising, causing this to become less concentrated or more dilute. Okay, so until the two sides have an equal concentration, the water will move across that membrane. That's osmosis, movement of water. Okay, so grab your self-check. That's as far as we're going to go today. 1, 2, 3, 4 questions. I'm going to give you a minute to review this. This lecture would really lend itself to flashcards and a concept map. So 2 differences between active and passive transport. Hopefully you included energy in one of your answers. So active transport requires energy, passive doesn't. Hopefully you included the concentration gradient. So active goes from low to high, it climbs the hill. Passive goes from high to low. The difference between simple diffusion and facilitated diffusion is that you should include a protein in your answer. Facilitated diffusion requires a channel protein, a carrier protein, a transport protein of some sort. Simple diffusion does not. Osmosis should have something to do with water. So diffusion is any molecule across a membrane or in a space. Osmosis is specifically the movement of water across the membrane. 3 physical characteristics. If you go back to slide 6, you'll see temperature affects the rate of movement. Pressure affects how closely packed molecules are, and so does concentration. The steeper the concentration gradient, the faster molecules will move. Do you need any of those repeated? Number 2, the difference between passive and active, or passive, facilitated and simple, right? Simple diffusion does not require proteins. Facilitated diffusion does.
Could you do the last one?
Yeah. The last one is going to be temperature, pressure, and concentration gradient. And you can play that video out if you want. Any others you need? Okay. So I think this PowerPoint, if you want to do some studying, take this PowerPoint, look through it again, watch all the videos and animations, make flashcards, start creating a concept map. We're going to be adding on to this. We're going to do a day of osmosis and a day of active transport. So you can start this concept map with your diffusion portion, and then we'll add on to it as we go. Okay, so that would be a couple study tricks you can do. So in the next hour, maybe give yourself a 10-minute break. This was a good long time. I think we might have gone close to 50 minutes. So give yourself a 10-minute break if you need it, and then come back, do the virtual lab. It's a handful of slides and a 5-minute video. It won't take you too long. You should be able to get it done in the hour, okay? So does anybody have any questions for me now? Okay, so if you want any individual help, you want to stick around in case you need help later, you want to revisit me, what have you, you can do that. Otherwise, you are free to go. Thanks for coming.